Hi, and welcome to Concepts of Biology, Chapter 3. We're going to go over cells, their structure, their function, and pretty much everything you need to know about cells for this course. So, our learning objectives for at least the beginning part of this chapter: describe the roles that cells play in organisms. I'm going to tell you right now it's super important. Compare and contrast light microscopy and electron microscopy. And then summarize the gist of cell theory. Okay. We talked a little bit in our previous chapters about how living things are organized, right? We start with the atom, the building block of matter. Which then forms into molecules, macromolecules, organelles, and then eventually cells, which are the building block of biology. Those cells work together to form tissues, tissues functioning together form organs, organs into organ systems that make up a whole organism. A cell is a very similar kind of makeup. An animal cell will have some different things that we call organelles. Organelles you can think of as your cell's organs that help them do their daily functions of living. Some important ones: the nucleus, the cell membrane, actually they're all important, mitochondria, Golgi apparatus, cytoplasm, and we'll talk about all of them in a little bit more detail later on in the chapter. The human body has a bunch of different types of cells. All of these different types of cells work together to allow us to complete our daily activities. Your brain has nervous cells. Your liver has cells, they are called hepatocytes. Your muscle cells, your intestinal cells, your red blood cells, all kinds of stuff. What we're seeing here are some images of cells underneath a microscope. The first one is from a nose. These are specifically goblet cells. And they make mucus and all that stuff, but that's a conversation for a different time. These ones here are plant cells. And you can see the difference between the animal cells and the plant cells. The plant cells have a more uniform shape to them, and we'll talk about why that is. And then this third one are bacterial cells. And notice they don't look quite like either of them, and we'll talk about that later on. What you're seeing here are some microscopes. Microscopes are what we use to see all of these teeny tiny small things, because you can't see a cell with your regular naked eye. So there are two main types of microscopes that we use in general biology labs. The first is a light microscope. That's this guy right over here. And that's probably the one that you're most used to seeing in these labs. We have them in our classroom. We have them all over the place. The other one is a dissecting microscope. And that's used for bigger specimens, rocks, things that we want to see the surface of a little bit more closely. The light microscope uses slides, and the dissection microscope doesn't necessarily need a slide. Okay. These are just some images of things underneath a microscope. This first one is a salmonella bacteria, and then the second one is a scanning electron micrograph, SEM, which is a very specific type of microscope, and that one looks really, really, really deep in. So this is a very high-powered scope. And then some more things under a microscope. These are cervix cells from the uterus seen under a light microscope. This is more commonly what you would see. Cells under light microscope. Okay, great. So that's kind of how we view these teeny tiny cells and how we learn more about them. How did we learn about them in the first place? Cell theory. Cell theory was initially initially introduced by Antony van Leeuwenhoek. He was a biologist in the early phases of science, and he observed protists under some different lenses and saw that there was more that could be seen. The next major contributor, Robert Hooke, he coined the term cell. Cell just means a small room. In 1665, he published a paper that was looking at different cells, cork tissue through a lens, and discovered the bacteria and protists. Okay. And then in the late 1830s, a botanist, Matthias Schleiden, and a zoologist, Theodore Schwann. I would know these names: Matthias, Robert, Antony, all of these guys. I would know those names pretty well. And they proposed a unified cell theory, which basically stated that all living things are made of one or more cells. And they proposed essentially that So the unified cell theory essentially just states that all living things are made of cells, cells are the basic unit of life, and that all cells arise from existing cells, so they don't just pop into existence out of nowhere. So the next thing that we are going to do, we're going to talk about prokaryotes versus eukaryotes. We touched on this a little bit in lab last week, but we're going to be able, by the end of this, you should be able to name a few examples of prokaryotic and eukaryotic organisms, be able to compare and contrast these different types of cells, and then describe their relative sizes of the different types of cells. All cells, all of them, prokaryote, eukaryote, all of them, all cells have 4 common components. The first, an outer covering that separates the cell's interior from its exterior environment. That is called the plasma membrane. Okay, the plasma membrane. The second common component is a jelly-like substance within the cell where all of the other cellular components are found, and that is called the cytoplasm. That's this little jelly liquid on the inside there. The third is DNA. DNA is the genetic material of the cell. Can't spell, sorry. And then finally, the last thing that all cells have is ribosomes. Ribosomes are the organelle that synthesize proteins. Remember, this is my little shorthand for protein, because I don't want to write out the whole word. Okay? So all cells, prokaryotic or eukaryotic, will have these 4 things: a plasma membrane to keep the outside in, a cytoplasm to hold all the inner stuff, DNA to give instructions, and ribosomes to make proteins. There are two types of cells. Prokaryotic cells, you'll know them by their lack of nucleus, by their lack of organelles, and that they are usually bacteria. Okay, so prokaryotic cells are not going to have a nucleus. They're not going to have organelles. Usually your prokaryotic cells will be bacteria. Eukaryotic cells will have an organelle, will have organelles and a nucleus. And usually they will be plant or animal cells. And animal cells. Okay. Those are the main differences between those two. And we'll talk a little bit more about those. So when you're looking at a prokaryote, like I said, doesn't have any organelles. No nucleus. The DNA is just kind of hanging out here in the center in what we call a nucleoid. Don't get those confused. But it doesn't have any of the mitochondria. It doesn't have any Golgi apparatus. It's got nothing, okay? So it just has that cell wall, the cytoplasm, and DNA, and some ribosomes to make proteins. That's all prokaryotic cells need to do. They are pretty simple. Whereas your eukaryotic cells are much, much busier, okay? A eukaryotic cell will have a lot more things going on. Your nucleus has your DNA, it has a nucleolus as well. All these different types of organelles that do different functions, okay? It's very important to note that with eukaryotic cells, the form will follow the function. Okay. That's super important throughout all of biology. Form follows function. You're going to find that all over the place. So in both animal and plant cells, the eukaryotic cells are a lot busier. They've got a lot more going on. Okay. This diagram is just kind of showing you the differences in cell sizes. Remember how we talked about how big a gram actually is. Right now we're functioning in meters, but if a human being is 1 meter and a frog egg is 1 millimeter, the size difference is incredible, right? So the differences in between these sizes shows you how big versus how small a cell really could be. So bacteria is probably on the smallest ends of things that we can see with a light microscope. And when we're getting into proteins, lipids, stuff like that, we can't see those with a light microscope. We would need a stronger, higher powered microscope, such as an electron microscope. So let's get a little bit further into the specifics of eukaryotic cells. We're going to talk about the structure of eukaryotic plant and animal cells. Plant and animal cells. Sorry about that. We're going to state the role of the plasma membrane, its functions, what it does, how it helps the cell. We'll summarize the functions of the major organelles, as well as describe the cytoskeleton and extracellular matrix. The plasma membrane is made up of a phospholipid bilayer. Remind me in class to show you guys my phospholipid bilayer hand signal, usually with some proteins embedded in it. These proteins help with all kinds of things, transport, with signaling, with anything that you could think of, a plasma membrane with proteins embedded will help a cell accomplish those things. The phospholipid bilayer is made up of these phospholipids that have a hydrophilic head and hydrophobic tails. Okay, so the head will be out towards the surface, touching more water, all of those things, because it can handle it, while the inside tails will kind of turn on their head so that they are protected from that liquid. So you got heads on the bottom, you got heads on the top, and you have tails in the middle. Okay. There are other things in your plasma membrane like cholesterol, carbohydrates, other proteins, things like that. Okay. You can also get some finger-like projections called microvilli, which helps to increase surface area, and that's important in places like the stomach, the intestines, things like that. Okay, so the phospholipid bilayer, plasma membrane, I will kind of use them interchangeably, but plasma membrane helps to keep ins in and keep outs out. Does a pretty good job of keeping what needs to be in, and what needs to stay out, out. Okay. The cytoskeleton, made up of 3 different kind of parts to it, are the microfilaments, the intermediate filaments, and the microtubules. Okay, they all make up the cytoskeleton. Microfilaments are the smallest. Intermediate is mid-sized, and the microtubules are large. And they just help with keeping the cell upright, for lack of a better word. They are the internal structure of the cell. These extra little bits that you see on the cells, these help with movement. Flagella are a whip-like tail that helps to just kind of move the cell as it needs to go. Okay. Cilia are little hair-like projections that also help with movement. I like to think of cilia as like millipede feet, and then flagella as just like a regular tail for moving. Okay. Again, in eukaryotic cells, there's a lot going on. There's a lot of organelles, but we're going to go through them bit by bit. Okay, so the first one that we're going to talk about is the nucleus. The nucleus is this guy right here. So the nucleus is a part of what we call the endomembrane system. Okay, and the endomembrane system is basically just a group of membranes and organelles that work together to help the cell do what it needs to do. Okay, the nucleus, probably one of the most important organelles in the cell. The nucleus you can think of as the brain of the cell. It is what holds all of your DNA, which is your genetic information, genetic instructions that your cell needs to be able to do its daily functions. There's a couple of layers to the nucleus. The first one is the nuclear envelope. The nuclear envelope is double membraned. And it is made out of that phospholipid bilayer. Okay. The nuclear envelope has what we call pores. Those pores help to control the passage of ions, move things in and out. Okay. The nucleoplasm, very similar to the cytoplasm. It's just that jelly-like substance in the nucleus. All right. Inside the nucleus, you'll see chromatin. Chromatin is the uncoiled version of DNA, chromosomes. Okay. And you usually only see those fully formed chromosomes right before the cell is about to divide, right before it's about to go through mitosis or meiosis. Okay. And then the last main part of the nucleus that we like to talk about is the nucleolus. The nucleolus is where ribosomes are formed. Okay. And then those ribosomes are then transported out of the nucleus via those nuclear pores. Okay, so now we've talked nucleus. We just finished talking about the nucleus, the nucleolus. That's this guy right. Oop, not all of that. That's this guy right here. I'll outline him again in red. Now we're going to talk about the endoplasmic reticulum. The endoplasmic reticulum usually sits just outside of the nucleus. Sorry, I'm trying to make it into a color that you guys can actually see. But it's this little like kind of bridge looking thing just outside of the nucleus. See, like I said, just outside the nucleus, they are right on that other edge. Okay. The endoplasmic reticulum is a series of interconnected membranous tubules. Connected tubes. Okay. They have two main functions when we talk about the endoplasmic reticulum. We talk about the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Okay. The inside of those tubes, we call them lumen. And it is another phospholipid bilayer. Probably in future for phospholipid, I'm just going to draw these guys. Okay. The rough endoplasmic reticulum is called that because ribosomes stick to its surface and make it look kind of rough or studded. Okay. These ribosomes will synthesize proteins while attached to the endoplasmic reticulum and transfer those proteins into the lumen so that they can go through other modifications and folding, anything like that they may need to go through. Okay. The smooth endoplasmic reticulum does not have ribosomes. And the smooth endoplasmic reticulum, sorry, I'm going to bring these down here. The smooth endoplasmic reticulum will usually help with synthesizing like carbohydrates, lipids, some steroid hormones, and does a little bit of detoxification. Okay. But the main thing that you need to know is that there's two types of endoplasmic reticulum, rough and smooth. The rough has ribosomes, the smooth does not. They both synthesize things. This one's rough, this one's smooth. They're named almost purely for their appearance and their function, but that's a different conversation. Okay? So now we've talked nucleus, we've talked endoplasmic reticulum. Now we're going to talk Golgi apparatus. The Golgi apparatus, also known as the Golgi body, is responsible for sorting, packaging, tagging, and shipping molecules, cell contents, wherever they need to go. Sort, tag, pack, modify, and ship cell contents. You can kind of think of the Golgi apparatus as your cells, Amazon shipping plant. Okay. The Golgi apparatus has vesicles. Vesicles are just these small kind of bubbles. Okay. These small little bubbles to help move content. They're called transport vesicles more specifically. Okay. So things will come in on one side of the Golgi apparatus and then exit through the other. And it has a trans face and a cis face. One of them faces closer to the endoplasmic reticulum, the other is on the opposite side. Right. So nucleus, cytoskeleton, we talked plasma membrane, Golgi apparatus, we talked endoplasmic reticulum. Now let's talk about lysosomes. Lysosomes, right here. Okay. Lysosomes will function as your cell's garbage disposal. This first root, lys, L-Y-S, means to break. And so lysosomes will use digestive enzymes to kind of break down any of the proteins, polysaccharides, nucleic acids, even old organelles. Basically anything that isn't working anymore, anything that doesn't need to be in the cell, the lysosome will take in via this process called phagocytosis. This root phago means to eat. We'll take in that substance via phagocytosis, start to digest it, and then pinch itself off and break it down and make it disappear. So lysosomes, your cell's garbage disposal. Okay. Vesicles and vacuoles. Vesicles and vacuoles are just membrane-bound sacs that help to store or move things. Okay. Vacuoles are large vesicles. And it doesn't necessarily, and the membrane doesn't fuse with other membranes, it just kind of stores the things themselves. Okay. But you can think of both of these as bubbles for transport and storage. I don't know about you, but when I think about vesicles and vacuoles and things like that, I think about how Glinda, the Good Witch in Wicked, moves around with her bubble and then pops it so that she can do whatever it is that she wants to do. That's what I think of when I think of vesicles and vacuoles. Just a little aside. Okay. Next, we'll talk about mitochondria. We all know mitochondria's go-to phrase, powerhouse of the cell. But the mitochondria is actually so much more than that. They're fascinating little organelles that do a lot for our body. For now, all you need to know is that they are the location of the electron transport chain. Electron. And that electron transport chain is what makes all of our ATP, which is our body's energy currency. ATP is just the acronym for adenosine triphosphate. Sorry, I know it doesn't really fit. Okay. The mitochondria has a double membrane, an inner membrane and an outer membrane. You can kind of see it here. Let's do this in red. Outer membrane and inner membrane. Okay, so it has a double membrane existence, and then this space in between, we call this the intermembranous space. Okay, and then on the inside of that inner membrane is the mitochondrial matrix. Just keep that in your brain for when we start talking about glycolysis, energy production, all of that stuff. So animal versus plant cells, we kind of jumped a little bit, but an animal cell is usually about 10 to 30 micrometers, and a plant cell can range from anywhere from 10 to 100. Okay, and if you remember from some slides ago, the animal cells were more circular than their plant cell counterparts that were a little bit more cubic or rectangular. Okay. So, some things that animal cells have that plant cells do not. The first, I don't want that color. The first would be centrioles. Centrioles are going to be an important part of mitosis later on. The next would be centrosomes. Again, another important mitosis component. And then third would be lysosomes, those garbage disposal organelles. Plant cells have some things that animal cells do not. Plant cells will have a cell wall. Animal cells have something, I won't even say similar because they're quite different. In animal cells, it's a plasma membrane. And they're not exactly the same thing as a cell wall, and I'll explain why a little bit later. Plant cells will also have chloroplast. That is what keeps them green. And then plant cells will also have a large central vacuole. In this case, that vacuole kind of acts like a garbage receptacle. This is a chloroplast. This is the gist of what a chloroplast looks like. Chloroplasts have their own DNA. I also forgot to mention mitochondria also have their own DNA. That's important. We talk about that because, well, I'll explain why later on. But chloroplasts, very similar to mitochondria, have their own DNA. And ribosomes, they are useful in creating energy and making substances that the plant will need to survive. Okay, so chloroplasts are somewhat, hear me when I say that, somewhat functionally related to mitochondria. Okay, they both have that double membrane thing going on and a lot of stuff on the inside that helps to create and make energy. The extracellular matrix is exactly what it sounds like. The extra meaning outside the cell matrix is just secreted substances by the cell. Sometimes it's proteins, glycoproteins, collagen, all kinds of things. Okay, so the extracellular matrix kind of holds the cells together to form a tissue, but also allows for some communication. That's communication. So all of the things that that get secreted by that extracellular matrix, those glycoproteins, those collagen molecules, all of that stuff are all used to signal to other cells. Glycoproteins are used. I'm not going to be that broad, but glycoproteins can be signaling proteins. Things like that. So there are 4 kinds of connections between cells. The first is a plasmodesmata. Sorry, a plasmodesma. I've always called it a plasmodesmata, but I digress. A plasmodesma holds 2 plant cells together and kind of acts as like a channel to send components through. Okay. A plasmodesmata will kind of, what's the word I'm looking for? Join the cytoplasms of the two cells. Okay, so that's number one. That's this top guy. The second is going to be a tight junction. Okay, and a tight junction, this is a watertight seal. So this is a watertight seal. It doesn't really let anything go. The only things that pass through them are things that specifically pass through those channels. You're going to find these kind of in epithelial cells, epithelial spaces, and they prevent leakage. Okay. Those tight junctions are in animal cells usually. Okay. The third type of cell is called a desmosome. Desmosomes are found in animal cells, and they kind of act as like a spot weld. You know, when you're trying to put two pieces of metal together and you use that little thing that ignites and sends sparks everywhere. It's kind of like that, right? So it joins the epithelial tissues cells together in kind of a sheet-like formation. Okay, so they still hold things together. They just do it a little differently. And then the last is going to be a gap junction. Gap junctions are very similar to the plasmodesmata, in which they have channels for things to pass through. They are different, gap junctions and plasmodesmata. Gap junctions, you're going to find these in animal cells as well. Okay. They do more than just transport liquid. They transport ions. They transport all kinds of different stuff. So for this next section, we're going to be talking about the cell membrane. We kind of touched on it a little bit. It's that phospholipid bilayer that keeps the insides of your cells in and the outsides of your cells out. So we're going to understand the fluid mosaic model and then describe the function of phospholipids, proteins, and carbohydrates in membranes. So we talked a little bit about that fluid mosaic, or that phospholipid membrane. The fluid mosaic model basically just states that all of these structures within the cell membrane are fluid, movable, and still will maintain integrity. Okay. The fluid mosaic model was proposed in 1972 by S.J. Singer and Garth Nicholson. And essentially, like I said, they just proposed that the cell membrane is something that kind of moves and adjusts as the cell needs. It is a fluid, moving, breathing kind of piece of the cell. It's primarily made up of that plasma membrane, proteins, some carbohydrates, glycoproteins, glycolipids, and other kind of signaling molecules. The cholesterol acts like antifreeze so that your cells don't become entirely rigid when it's cold and all that stuff. Like I said, the heads are polar, and the tails are nonpolar. Because those tails are made up of fatty acids. Okay. Integral proteins are exactly what they sound like. Integral proteins are integrated into the cell membrane. Okay. They will usually span the entirety of the cell membrane. So they will have one end going out the outside, one end coming in. Okay. Peripheral proteins are usually just hanging out on one side. So this guy here is a peripheral protein. He's just on one side, just hanging out. Okay. A protein channel will usually allow for some specific ions, anything like that. The whole point of the cell membrane is to keep what needs to be in, and keep what needs to be out. And the cell will determine what it needs and what it doesn't. So this is just kind of talking about how viruses will infect specific organs. All of those glycoproteins, those glycolipids, can be very specific, or they are usually very specific, right? And so a virus will have a specific piece to it that can bind to a glycoprotein. In this case, with the HIV, it's a CD4 receptor that allows it to enter into the cell. Because as much as our cells try to keep bad things out, they are only responding to the mechanism that they know. So if there is, on my cell, say there is a receptor that is shaped like this, that fits like this, whatever, and usually it has my good, it'll be green because it's good, my green good molecule that then starts a pathway down into my cell that is good and beneficial for me and for my health. Say my virus has learned what this receptor is, it has bounced around other humans, found that this is what the receptor is, and then will make itself fit into that receptor, but be bad for my cell doesn't know the difference. It's just going on the receptor and the receptacle. Okay? So that's how that specificity allows for kind of movement. Okay. Now we're going to talk about transport. We'll talk about why and how passive transport happens, the process of osmosis and diffusion. We'll talk tonicity and its relevance to kind of passive transport. Okay. The cell membrane is known as what we call selectively permeable. By selectively permeable, we just mean that it allows some substances through and does not allow others. Some things will pass through easily and some things will not. Okay. Remember, our cell membrane has that phospholipid bilayer. So there will be some things like polar molecules that will have an easier time kind of getting through versus other non-polar molecules. Okay. So some things that will pass through easily would be things like fat-soluble vitamins, those are like A, D, E, K, some fat-soluble drugs, all kinds of stuff like that. molecules of water, CO2, are small enough that they will pass through relatively easily. Sorry, not water. CO2 and oxygen will pass through relatively easily. Things that will be a little bit harder to pass through are polar substances, charged ions, etc. Okay, so charged ions is stuff like sodium, potassium. I think potassium is also 2+. Calcium, I know calcium is 2+. Chloride, stuff like that. Anything that has a charge to it will be a little bit harder to pass through. Sugars, amino acids, etc. When we talk about movement through a cell, there's a couple of different ways that we talk about it, okay? The first thing that you really need to know about is diffusion. Diffusion, by definition, is just a tendency for a substance to move from an area of high concentration to an area of low concentration until it is equal. What does that mean? If I had a box, and that box was full of, there's a little divider in the box. So that box had a bunch of circles on one side and a bunch of squares on the other side. This is not even. This is not what we would like to see. The squares don't necessarily like being around the other squares. The circles don't necessarily like being through the other circles. So they will go through a process of evening out, adding a couple from one side to the other until they are completely even. That is what we call diffusion, an area of high concentration to an area of low concentration. And we'll talk about that further in labs as well. Okay. So over time, things will move into an area of equilibrium. Osmosis is just the specific diffusion of water. That's all. Water will always move. Water, remember this, water follows solute. The water will always move from an area of low concentration to an area of high concentration to maintain that kind of balance. There are several different things that will affect the rate of diffusion, things like the mass of the molecules that are diffusing, the temperature, the density of the solvent, all kinds of stuff. Because of that water following solute, we have something called osmotic pressure. And when the osmotic pressure of a cell changes, it will force the cell to be either hypertonic, meaning that all of the water has left the cell because my environment is super saturated with solute. Okay, so my cell is in a hypertonic solution. Isotonic means that everything's chill, everything's dandy. We are passing water in just as much as we are passing water out. Okay? And then when your cell is in a hypotonic solution, meaning that most of the solute is inside of my cell, the water is going to travel into the cell and eventually burst it because it's too full. So water will always follow solute. Same thing in plants. That central vacuole helps with what we call turgor pressure for a plant cell. In animal cells, it's osmotic pressure. In plant cells, it's turgor pressure, and it's the same thing. Another type of movement through a cell that we talk about is facilitated diffusion. Facilitated diffusion essentially just uses one of those transmembrane proteins, one of those proteins that has an opening on the outside and an opening on the inside, to move molecules through from an area of high concentration to an area of low concentration. Okay. Facilitated just means with help. But it's that same process of moving solutes from high concentration places to low concentration places. And then the last thing that we're going to kind of talk about today is active transport. Active transport is very similar to passive transport. The difference is that active transport uses ATP. All of the diffusion that we were talking about before does not use any energy to move the solutes. Active transport does. So we will understand how electrochemical gradients affect ions. We'll describe endocytosis, including phagocytosis, pinocytosis, and receptor-mediated endocytosis. And then we'll also talk a little bit about exocytosis. All just fancy words for cells eating, drinking, and pooping. So the electrochemical gradient is it's basically a way that we understand the movement of ions through a cell. The inside of a cell is generally negatively charged, and the outside is a bit more positively charged. So the gradient will want to move some of the high charged potassium ions that are at a higher concentration inside the cell out of the cell. And to do that, it has to use some of these proteins, these integral proteins, to kind of move the charges as they need to be. These electrochemical gradients are really important in things like muscle cells, nervous cells, stuff like that. This is your sodium-potassium pump. Okay. So this electrochemical gradient goes against what we think of when we are trying to balance a cell. So to move ions against their concentration gradient away from what they would want to be in, we do something called active transport. Okay? Active transport uses ATP to push molecules against their concentration gradient. So in this diagram, you can see that there is a high concentration of potassium on this side inside the cell. So we're using ATP to take any molecules of potassium that we have outside the cell and pumping them into our cell, even though there's already a high concentration of potassium inside the cell. So for every 2 potassium that come in, we kick out 3 sodium molecules. Okay? And so we use that ATP to move against our concentration gradient. Other types of movement through the cell, when we have to move kind of large molecules or any of the external fluid, we do a process called either endocytosis or exocytosis. There's a couple of different types of endocytosis. The first is phagocytosis. This is cell eating. The second is penocytosis. This is cell drinking. And then receptor-mediated endocytosis is a specific receptor will form kind of a vacuole. This is just specific eating. Almost like your cells on a diet. Not really. It's basically just if something hits that receptor, it will form a vacuole. It'll pinch itself in and then move in or out of the cell depending on what it needs to do. And then exocytosis, bringing a vesicle, usually sent from the Golgi apparatus, the smooth endoplasmic reticulum, any one of those places, to the cell surface, and then letting all of the contents of it go. That's exocytosis.